Hello and welcome back. Today we're going to talk about plant anatomy and the effects of plant anatomy on extractive techniques. By the end of the day, you should have a fairly deep understanding of where plants store the natural products and the ramifications of this on extractive techniques. The key concepts that you should take away are being able to identify major plant tissues, recognizing secretory structures, why different plants contain different compounds, predict extraction challenges from plant structures, and connect storage locations with extraction methods. To start out, it's important for us to understand the major structures of a plant. These include flowers, fruits, leaves, roots, and stems. If we look over here, we can see where these grow. So we have starting, excuse me, at the bottom, we have our roots, which include the primary root or tap root. We have lateral roots, which come off of the primary root. We have root hairs, which come off of the both primary and lateral roots, and these are helpful for grabbing local nutrients. You have the root tip and the root cap, and these are going to drive the growth of the plant forward. Above the ground, we have, excuse me, we have the shoot or the trunk. This is sort of what we see as the major component. It's composed of ground and vascular tissues as well as dermal tissues, and we're going to spend some time talking about that in a second. But this trunk is the major structure here, and we have leaves which come off of the stem. Depending on the progression through its life cycle, our angiosperms are going to produce flowers, which will eventually mature into fruits containing seeds, like we talked about last time. The outside of the plant is going to be covered in its epidermis. And then we have several additional minor structures, which aren't very important for us. And these include nodes and shoot tips. So these nodes are places that new leaves or flowers can form, and the shoot tip is what's going to be responsible for driving additional growth of the overall plant. Okay, so now that we have a broad overview of the types of plants, I want to talk briefly about the hierarchy in structures that we have in plants, and you know this extends, of course, to other life kingdoms such as animals and fungi. So we start with an organism, which is a system of organs, and those each have different organs. So, you know, like a leaf would be a plant organ. That organ is made up of different tissues. So these are like type cells that are organized in a fashion for a specific purpose. Those tissues are composed of individual cells, and those cells have different organelles in them that do specific tasks. So let's go back and think about tissues and cell types. So in terms of organs, we have, for plants, we have roots, stems, and leaves. These are going to be our major types of organs. Sorry, we also have flowers if we have angiosperms. And these can be broken down into tissues. There are three major tissue types in plants: ground tissue, vascular tissue, and dermal tissue. And these are made up of different types of cells. Okay, so we're going to start with our cellular structure of plants. The key is for you not to memorize a plant cell structure, but just be familiar with the major components that are going to have impacts on us. So the major component of a plant cell that makes it a plant cell are two features. One being a cell wall. So this is unlike animals, which do not have a rigid cell wall. They just have a, flexible membrane, versus plants are going to have this rigid cell wall composed of cellulose and hemicellulose and lignin. Inside the cell, inside the plant wall, you have several major structures. You have the nucleus. So this is where the DNA is held, the nuclear membrane, which protects that. You have the cytoplasm, which is going to be sort of the liquid sitting within the cell. You have the chloroplasts. And so these are what are the structures that are going to convert sunlight into sunlight, CO2, and water into sugars. You have mitochondria. So these are these purple, illustrated here as these purple cells, purple organelles. And these are responsible for sort of the backwards reaction, converting sugars into the usable form in the cell, which is ATP. You have Golgi bodies and smooth and rough endoplasmic reticulums. So these are responsible for construction and repairs of proteins and enzymes. And then we have this interesting structure over here, which is also lacking in animal cells, called our large central vacuole. This organ is a fluid-filled organelle, and it's going to expand and contract in order to control the osmotic pressure within the cell. Because that cell wall is rigid, the cell, if it loses water or gains too much water, cannot expand and contract. So this vacuole is there to maintain the pressure as in the cell, and then that can help either expel excess liquids or draw in resources from the vascular system as it expands and contracts. Okay, so now let's take these tissues and these cells, and these cells are going to organize themselves into tissues. As I said before, there's going to be three major tissues that we will be talking about: ground tissue, vascular tissue, and dermal tissue. So we're going to start with the ground tissue. Its definition is pretty simple. It's defined as everything that is not vascular or dermal tissue. This tissue acts as a structural support storage mechanism, helps with various metabolic activity and photosynthesis. There are three major types of cells associated with the ground tissue. The first, shown down here in a micrograph. Here we've taken a slice of a tissue and used staining so that we can see different pieces of the cells. This is the collenchyma cells. These act to support new growth. These cells are alive. They form elongated tubes, and they have, as you can see, sort of uneven and relatively thick cell walls. And so as the plant grows, as there's new growth, we start out with these collenchyma cells. As they further grow, and the plant continues to grow and needs a more rigid structure, these cells die off and form sclerenchyma cells. So unlike the collenchyma cells, these cells are actually no longer alive. So they're no longer actively respiring or undergoing photosynthesis. What they do, though, is they're forming these elongated structures. So here's a cross section in one direction. Here's a cross section in another direction. These cells in red are these sclerenchymal cells. And you'll see that they form these long tubular cells in nice sort of patterns. They're no longer, as I said, they're no longer undergoing active respiration or photosynthesis. And they act to support the system, support the tree. They're really there as a support system. What you'll also notice is, in comparison to the collenchyma cells, they have very, very thick structures outlining the cell. So you can see here, out there. So these are actually secondary cell walls, and they're very thick and rigid, and so they act to as essentially the bones of the plant. The third type is the parenchyma cells. These are alive. They tend to be spherical. They have thin walls, and these are responsible for photosynthesis and storage. So these thin cell walls facilitate the transport of material into and out of the cell so they can undergo respiration, photosynthesis, etc. Okay, so the key thing is just to sort of, not to memorize these different cell types, but just know that there's sort of, these dead cells which act as support. We have alive cells which are for growth, and then we have cells that are going to be our active photosynthesis cells. Similarly, we have, or the next type of cell that I want to, or tissue that I want to talk about is vascular tissue. And so just like the human vascular system, the vascular tissues in a plant are responsible for transporting the different nutrients through the plant. There are two major types of within this tissue, zymal and phloem cell tissues. So the zymal tissues is responsible really only for the flow of water and minerals. So this is not containing sugar. This is not containing the proteins, enzymes, essential oils, right? This is just a water and mineral transport system. And so it elongates from the root and transports up to the top of the plant. Depending on the type of plant, they can be located in different portions, but tend to be located towards the center of the plant. They form these larger pores through the structure. Additionally, they tend to be very stiff, and they use lignin, which are sort of rigid molecules, to form them. And we don't have walls between the cells. The walls have broken down, so we form these continuous tubes. Conversely, we have the phloem cells. So these undergo two-directional flow. So material can move up and down, in and out of through these vessels. And this is because we're moving not just water and minerals, but we're moving water and sugars, other small molecules through the plants, right? Because the leaves, which are sort of the engine of the plant, right, are needing to share the sugars and other molecules with the rest of the plant. So we have stuff that goes everywhere. These tend to be much smaller, so we can see in this structure over here, much smaller cell types or structures than we did with the xylem. Yeah, and so this is where we're going to get, tapping sort of in terms of sap collection, et cetera, are coming out of these structures. Okay, so a little bit more, just like in the ground tissue where we had cells which were both alive and dead, this also exists in these vascular tissues where the xylem contains mostly dead cells, while the phylum uh, is going to contain mostly living cells. Right, and so we can sort of look at this and say that, what's the key difference? Well, the xylem is going to transport water and minerals, while the phloem is going to contain the sugars and transport the sugars. And so the direction is up versus up and down, and there's a lot more biomass in the transport of water. Again, there's an entire world and many lectures in the full botany class, which could be spent talking about these different structures in detail. But this should be enough for our class. The last type of tissue is our dermal tissue. And this is the very outside piece of the cell. So if I take, here's a leaf, and I take a cross section of this leaf, we see these dermal tissues are just this very thin layer up top. All this we have our upper epidermis and cuticle. So these are our dermal tissues, but this mesophyll consisting of the palisade and spongy tissues and vascular tissues, right? That's our either vascular tissues or ground tissues. It's just this very thin layer up here. And these tissues are going to contain a few types of cells. So these are epidermal cells, guard cells, stoma complexes, and epidermal hairs, which are called trichomes. And we'll spend some time talking about trichomes later. We can see down on the down on the right, a picture of what the epidermal layer looks like. And we can see the two different types of cells, so are epidermal cells. So these are these square cells. And then we have these guard cells, which look more like bean shapes. And then we have what's called a stomatal pore. So this is a pore that enables gases and liquids to move through the leaves. So in reality, what we want is oxygen and CO2 to move in and out of this pore, and then water usually is coming out of this pore through transpiration. Some of these cells, of course, are going to produce trichomes, which are going to be one of the major sources of essential oils and other herbal products for us. So just to give an electron microscopic view rather than just a cartoon, we can see here's our surface. We can see the trichomes zoomed in. And if we look at the surface, we can see here's the stomatal pore, the guard cells, and then these plate-like cells being the epidermal cells. Okay, so now that we've talked about the different types of tissues, how these tissues grow tend to lead to two categories of plants that we can think about. So herbaceous plants and woody plants. So these herbaceous plants are soft with non-woody stems, and the woody plants obviously have woody stems. So that the epidermal layer forms a bark versus a more malleable layer in this in the herbaceous plants. Right? And so we can think about woody plants as being more things like trees or long-lived shrubs, and herbaceous plants being things that in general die each year, so they're often annuals, or the stem and leaves tend to completely die back each year. You'll also notice if we take a cross section of the stem or the trunk, the excuse me, the locations of our vascular system in the herbaceous plant is rather different than in the woody plants. So in the herbaceous plants, they tend to be distributed throughout the cross section, whereas in our woody plants, we end up having sort of the our dead structural ground cells in the middle. Then we have the xylem and phloem sort of situated near the edge, and then finally the bark on the outside. So if once again, considering the differences, the herbaceous plants tend to have soft green stems. And again, those parts generally die back seasonally, and they generally live for a shorter period of time compared to our woody plants, which are generally long-lived. The above ground parts persist year after year, and you've got that thick woody trunks and bark. When we think about what this means for us in terms of extraction, it's often much easier to get the essential oils and other compounds out of the herbaceous plants because we can grind them more easily. We have, less protective structures that are preventing that we need to break through to get at those oils. And generally, because they do grow, they're softer, they're faster growing, and we generally have shorter time to harvest. Conversely, in the woody plants, those harder tissues generally require much more grinding, more energy to grind and chip away at those structures. Some of those oils and other compounds that we're interested in tend to live deeper in the tissues, so therefore the extractions generally take longer because we need those compounds to move through all of this extra biomass, which is there to try to prevent those extractions from happening. And generally, of course, these have longer growing cycles, but you can, if you're careful, have sustainable harvesting because these plants aren't going to die year after year. So as long as this is done carefully and you don't harm the tree or the shrub, you can keep coming back. Okay, so we've talked about different plant organs and tissues. Like I said at the beginning of today's class, the different organs that we're usually interested in are things like leaves, flowers, flower buds, bark, fruit peels, and seeds. And so these have different examples. So, we can think of extracting mint and menthol from leaves. Chamomile is the flower. Clove is a flower bud. Cinnamon is the bark. Our fruit peels give us our orange oil, and then within fennel seeds, there's various compounds that act as defense mechanisms. Okay, so within these different structures, we generally have three locations where we store the essential oils and flavors and fragrances that we're interested in. We're going to put these in three categories: trichomes, oil glands, and secretory cavities and resin ducts. So here's an illustration of a trichome, an oil gland and secretory cavity, and then a resin duct. We'll take a second to look at each of these. So our trichome is a fine outgrowth of a hair-like structure which extends from the surface of the plant. And so here I've shown pictures of several different zoomed in plants, and you can see these hair-like spikes, and they come in different um, shapes and sizes, from very large on this sundew down to the microscopic, shown here for lavender. They can be sort of single hair or branching, and densely packed or more loosely packed. And these can be unicellular or multicellular outgrowths. So these trichomes generally are going to be excreting, we can think of them as glandular trichomes. So these are the trichomes that are going to excrete secondary metabolites. And these are often for chemical defenses. And so if we look at the edge of the epidermal layer, we have this cell here. It's a unicellular glandular example, and it's going to produce different oils, secondary metabolites, which are pushed up through this structure. And these are going to contain things like terpenoids, phenylpropanoids, flavonoids, ketones, etc. Don't worry about the names of these. We're going to talk about that next time. So we can look at these to bring some examples. The first one that I want to show is a stinging nettle. So here you have the stem of a stinging nettle, and then you have the trichome sticking out here. If you've ever been unfortunate enough to touch a stinging nettle, what's happening is sitting within this trichome are histamine active terpenes and other compounds, and it punctures these are structures that they form needle-like structures. They'll puncture your skin and leave your skin burning and itching for about a day. Hence the name stinging in stinging nettle. That's of course the common name. The scientific name is placed under here. These trichomes have evolved for many different purposes. Besides just purely defense, they can actually be offensive. So if we think about a Cape sundew, which is shown here, it's this pretty green stem with these red trichomes and sort of bulbous ends of the trichomes. This is a carnivorous plant, and so it's luring the various insects to come and interact with the trichomes. And then when they get touched, they're sticky, and it traps the trichomes act to trap the insect, and this whole structure will sort of roll up, and it will then eat the insect. And then, of course, we can have neither offensive nor defensive type mechanisms, shown here for our English lavender. This is an example of a branching trichome rather than these straight trichomes. So these branching structures can also be called dendritic formations. And this is where the lovely smell of lavender is coming from. Okay. We can also have non-glandular trichomes. So these are generally purely structural protective agents that are used to help minimize loss of water, regulate temperature swings, UV protection, and as an antibiotic and herbivore defense mechanism. So unlike our glandular trichomes, these are not going to produce those chemical agents, but they can store some. And so here you can see in Cystis salvifolius, if you look closely, there's trichomes on these leaves here. Okay, so let's move on to our secondary containment structure. So this is oil glands and secretory cavities. And there's two major sets. So these are uh, oil cavities in orange peel. So if we take this orange peel, we zoom in, um, we start having a cell which is going to form an oil gland. It replicates, forms this gland, and then eventually forms a, um, uh, a void within the tissue that these gland cells excrete or secrete their various compounds into them. And so then these oils, terpenes, etc., are going to be held in this oil gland cavity. And so you can see this here in a slide cross section. We've got the glandular cells staining a little red, and then you have the chamber left behind where the oil resides. And so this can exist both in places like the peels of oranges, and then also in leaves, stems, and other places. Here's an example of an oil cavity in a eucalyptus leaf. Again, you have these large structures that have formed, sort of voids where the oil is going to fill. Similar to oil glands, just as a comparison, you can get additional structures that look similar for UV protection. So this would be a pigment gland. It's going to look similar, but rather than storing oil, it's going to store pigments. And so think about things like, pretty plants might store reds or blues, purples, etc. in these pigment gland cells. The last structure that I want to talk about is a resin duct. This is going to be very similar to an oil gland or oil secretory structure, but rather than being a spherical structure that's very localized, this is an internal channel that's going to run significantly through the plant. So instead of just being a sort of a spherical structure, think about like a pipe. And so here we have on the right a cross section of a pine tree, and we can see these resin ducts within this structure. There's the secretory cells lining the pore or the duct. They're going to then fill that duct with the compounds. And so these are normally protective compounds, so that if the tree gets a wound, right, so it's cut or broken, this resin, this material will be exposed and can try to help cure and seal that wound. Okay, so if we want to think about these different structures, right, there's sort of three sets that we've talked about. Trichomes, which are these which store oil outside of the tissue. So this is my little drawing of a trichome sitting out, pushing out of a leaf. An example would be the mint family. We also have a lot of sets where we have oil in cavities in the glands, so they tend to be spherical and located within a tissue. So think citrus and myrtle families, myrtle being containing eucalyptus, where the desired oil sits within these spherical vessels. And then the last major structure is our resin duct. So these are, again, inside the tissue, but are long cylinders. And so an example would be pine resins and things like frankincense. Okay, so if we sort of pull all of these together, we can look at different organs what the chemicals are and what their functions are. So we have, for example, leaves, which generally we want those leaves because they contain trichomes. Um, an example being mint again, which we want the menthol, and its function is for defense. The flower organs are going to contain both trichomes and cavities. Additionally, some types of plants have what are called osmophores, which are rarer, and these are acting not so much as a holding location, but rather a active dispersal set. So these are not great for containing fragrances that we can harvest later because their purpose is to actually disperse the smell. These are here for pollinator attraction. An example would be rose oxide on a rose that gives it that rosy smell. If we think in terms of fruit peels, again, we have mostly cavity type structures. If we think about oranges and extraction of limonene, these are there as a defense mechanism to either prevent wounding, or if a wound does happen to have antimicrobial, anti-insectivore properties. Going into seeds, these generally form cavities, oil cavities. An example would be fennel. Within the fennel's oil cavities are anethol, and this acts as a antimicrobial, anti-herbivore set to protect the embryo within the seed. Lastly, sitting within the bark, we're generally after the compounds that sit within the resin ducts. Again, our example is cinnamon with cinnamol as the compound that we're generally after. And again, this is wound protection. Okay, so if we think about, if we pull everything that we've been talking about in this lecture together, we end up with this sort of diagram, which is going to show us, the major storage structures. So trichomes, oil cavities, and resin ducts. Where they're located, right? So the trichomes are going to be on the surface, so they're growing out of the leaves, stems, and flowers, whereas oil cavities are going to sit inside the tissue, and the resin is generally going to be found more in the stems, bark, and wood. And these are again inside their long tubes and channels, whereas the oil glands and cavities are more spherical. Because of these locations, how we can get at these oils is going to differ, right? So these trichomes are on the surface. So this, and they're not heavily encased. So they're easily ruptured. And this means that we generally have efficient release and access to these compounds. Whereas within oil cavities, we have to break open the tissue in order to get to them, and we have to then rupture this oil cavity in order to release the oil. So this is generally going to be a moderate release effort. Whereas to access resin ducts, these generally are deeper within the plant, generally has slow release, and so therefore it's going to take longer to get these materials out. So either we are going to take, it's going to require higher heat or longer times if we're doing things like steam distillation or um, other extraction, like solvent-based extractions, or we go through the wounding method where we actually make a wound in the tree generally so that these resins extrude and then we can pry it off. Right. If we look at the extraction approaches of the trichomes and oil cavities, steam distillation works usually very well with trichomes. We generally don't need massive grinding because everything is at the surface. Same thing with solvent extraction. Those molecules are already easily accessed. But in the oil cavities, we generally have to do some sort of grinding or maceration in order to rupture the cells um, in order to release that. So if there's one takeaway I really want you to have from this lecture today is, right, our plants that are making our natural products store them in very specific structures. Here are the structures that we've talked about: trichomes, oil cavities, resin ducts. How those, that dictates where they're located, and that is then going to control how easy it is to access those compounds. And therefore, we need to think about these extraction techniques, how we're processing them based on where these oils are being stored. Okay, so at the end of the day, you should understand and be able to answer the question of where plants store natural products and think about how that impacts extraction techniques. You should be able to identify major plant tissue types, recognize different secretory structures, understand why different plants contain different compounds, predict the extraction challenges based on the plant structure, and connect storage location with extraction method. For next time, sorry, that's wrong. For the next time, I want you to, we'll start considering what these molecules look like. So that's going to wrap up our second module on botany and plant biology. So you should know how we think about plant identity, metabolism, and reproduction, as well as where plants store natural products and how this impacts our approach to extraction. All right, I'll see you next time where we'll talk about the molecules themselves.